So if you don't mind, I'm going to get going. We're going back and revisiting one paper. Actually, I decided at the last moment to change up what I was originally planning to present to you, and I'm going to try to change it up in a couple of ways to make it rationally more approachable. So I'll go through that in a moment. And I want to go back and revisit epilepsy. I also want you as both genetics as well as neuroscience students to be familiar with different models that are out there. Some are genetic models, and we will come back to them. Some of these models actually give us some evidence about the genetics behind epilepsy, which is what we should always be looking for in animal models, and we'll come back to that later on in the course as well. But I wanted to revisit this idea of looking at different parts of your brain as a circuit, and in particular when a disorder like epilepsy occurs, and it can occur for a variety of reasons. It could be a viral infection, a bacterial infection, a hit on the head. You might be taking different drugs of abuse, or you might have a carcinoma that is pressing down on one part of your brain. You might develop epilepsy or seizure activity, and this is sort of an idea of a circuit. So I'm going to go back and tell you a little bit about different models. And in particular, I want you to concentrate today as we're going through. Some of it will be new, some of it will be a review for you about different models for developing seizure-like activity in mice, as well as in one model of a rat, which is actually much more surprising as well as much more exciting than we give it credence for. So I'm going to go back and revisit a little bit about seizures, give you some animal models, and especially the acute drug-induced models of seizure. A few years back in the neuroscience program, we actually used to have behavior. You worked with animal models where you did kindling, where you looked at epilepsy, and I was told that you no longer do those types of animal behavior models, which is a real shame because having an idea of how to handle animals is really important. And then I'll go through very briefly a genetic model that is incredibly exciting because it's a transgenic rat. You hear a lot about transgenic mice, but hearing about transgenic rats is kind of a neat thing as well. And I'm going to, instead of concentrating on two papers this week, I'm just going to concentrate on one. Part of the reason for this is, as you know, in the last 3 days, the surge of these great papers in neuroscience. And I'm not talking just like good papers. I'm talking this will forever change the course of how we look at neuroscience, how we look at it as a circuit, because actually, because of the genetic variants, using neurogenetics, by the way, the course that you're taking now in neurogenomic approaches, we now understand more about schizophrenia from this one paper than we have in the last 10 or 15 years. And we will be this paper, understand more about disorders, more about obsessive compulsive disorders. You'll understand why, for example, I'm interested in the onset fasciculus. If you are studying anything about anxiety, now you'll have a better idea as to why this is such a groundbreaking paper. So I'm actually reworking some of the lecture material. We will actually go back, look at stem cells next week, but the week after, we're going to start jumping into neurogenomics a little bit earlier. You won't be responsible for that paper, by the way, for your midterm, and I want to finish off on as much of the remaining things that you need to know as possible. So this is the paper that I'm going to talk to you about. It is not the first paper that came out. The first paper that came out on the circuitry of epilepsy was in 2009. It was part of the Dijsselroth group. This is actually a better paper in many ways than Dijsselroth's original 2009 paper and the follow-up papers in 2010 and 2011. This came out in 2013. I also wanted to take a step back because in many ways we talk a really good game about neuroscience, and we talk about all these great things that we're doing in neuroscience, and some of you have been asking some really good questions, and you should. As neuroscience students and as genomics students, you should be asking those types of questions. So let me ask you a really impertinent question without you looking it up on Google or anything else. Who is Jesse Gelsinger? Because, oh, yes?
You learned him about
Oh.
X. Wasn't he? Is he the guy? No, I feel like that's wrong. Yeah.
Take a shot anyways.
Was he the guy who died when he was treated with a vector with his immunodeficiency?
So this is the really cool thing. So you started off speaking really, really loudly, and then slowly over time, as you got less and less confident about, yes, that you had your hand. Do you have anything to add?
Was it ADA or I
Sorry?
can't remember. He was doing a gene therapy trial.
Yes.
He had like a mild form of something, and then he died.
Yeah. A lot of you have been asking a really fundamental question. Why do we use adeno-associated viruses? Why do we use adeno-associated viruses in the MDX model? Why do we use adeno-associated viruses in this paper? Why do we, in general, we talk about neurotropic, we talk about myotropic and how they like infecting different tissues. One of the reasons is actually from Jesse Gelsinger. Your colleagues are more or less correct, and I know some of you in neuroscience may not have heard it, but in 1999, Jesse Gelsinger was the first individual to actually die in a clinical trial because of therapy. And because of that, gene therapy has stopped for the last 15 or 16 years. You very rarely hear about gene therapy in humans, mainly because an immune response to the virus that he was being injected with, you still have to deliver that therapeutic gene, actually caused a severe immune reaction in Jesse. And we now know that that severe immune reaction was enough to cause him to die. And one of the reasons why we use AAV, because it has a very low immune response. So if you inject it into a rat, that rat or mouse will not have a severe immune response. And it's one of the reasons why we do the things that we do. And you should be always asking those types of questions. Yes, Dan. At the time, back in 1999, they were primarily interested in using 3 different viral vectors. We still use these, by the way. Adenovirus, not the adeno-associated virus, which is capable of replication, induce a large cytokine reaction. HSV is still used, but you know, for obvious reasons, you don't want to introduce that there. And parts of the HIV envelope. So the GP120 protein was commonly used as sort of a delivery system as well, way back in the day. And again, beyond sort of the purview of what I want to go through, but I want you to get a good understanding. Use the vectors that we do, and those of you who have been keeping up with the literature, actually the most exciting stuff I'm not to here today is actually what a specific receptor has been found for a virus to actually enter into a cell, so we can actually design even better adeno-associated viruses down the road. So I want you to focus on a couple of things throughout today's lecture. The first sort of background slides I don't start memorizing or going nuts with, and I want you to be thinkers, not memorizers. I want you to really learn to apply things rather than to try to memorize all these little details. You are in 3rd year. In 3rd year, you should be doing a little bit of both, to be quite honest. So we talked about epilepsy before, and we talked about how severe it is, has a number of different manifestations, from absence seizures, where you might be sitting beside someone who is losing consciousness, but not like closing their eyes and falling head down on the table, to myoclonic, clonic tonic type of seizures. Many devastating manifestations of this. Again, these are recurrent. You have to have more than 2 episodes of seizures to have a definition clinically of epilepsy. And right now, our options are very limited. So medications, lots of side effects, and people don't actually want to be on these medications. And when you stop having any type of seizure activity, you usually go off. There are forms of surgery that are available, and in some instances, for some types of focal epilepsy, electrical stimulation and modulation by electrical stimulation is a possibility as well. However, right now, we don't have a really 100% effective way to manage seizure activity. And medications are still the most common way in which people deal with these different types of epilepsies. And somewhere on the order of 20 to 30% of patients that take medications still have epileptic seizures. They're on the medication, they stay on the medication, it works, but they will still have the seizure activity. Yep, do you have a question, Kay?
For the recurrent seizures, though, does it have to be within a certain time frame?
No,
No. So
no.
even if you had a child.
Okay. So in most cases, for example, I told you in the last lecture, there are different types of seizure activities. So you might have it as a child for whatever reasons. It might be febrile, fever-induced, might be for a whole host of things, and it might disappear over time. However, if you do have another episode, as a child, it's unlikely after it's disappeared for a while, when you turn 18 to 21, that you'll have another episode. However, I'm gonna leave you with the thought when we get into one animal model, which is called kindling, and I'll leave you with that thought. So, is your brain actually more susceptible than someone else's brain that has never had a seizure? And I'll leave you with that thought when we get to the idea of kindling. So again, somewhere between 20 to 30% of individuals with epilepsy, even if they're on the medication that we have available now, whether it's a calcium channel blocker, a GABA receptor agonist, a GABA transporter, antagonist, whatever it is, they will continue to have epileptic seizures. And of the 20% that continue to have seizures, quarters of those individuals have a particular type of epilepsy, which is a focal. We're not talking generalized epilepsy. We're talking about focal epilepsy. It starts in one area of the brain and then spreads elsewhere. And if it's something that they should be looking at, their best option is not to actually be on medication. If it's not part of the eloquent cortex that you're learning about or anatomy classes, then surgery could potentially be the best option for them. However, it turns out that where these seizures start, and as all of you who have taken psychology know, that with HM it causes potential problems, especially in the medial temporal lobe, which is where many of these focal types of epilepsy occur. So we're really good at lots of things in neuroscience. In neuroscience, we're really good at being able to detect where sort of synchronized activity actually start. And we can map that out. We can actually go in, map it out, but taking care of that issue, though, is still a big deal. And so if the eloquent cortex, the part are involved in movement, speech, cognition, all of that, memory, and other things, are going to be damaged by that surgery or surgical resection, it becomes a huge issue for these individuals. And so looking at other types of options and other therapies is really, really important. There are, and this is a slide that I'm going to suggest that you, if you your second year neuroscience course, or you haven't had this in your psychology courses, or you haven't had it elsewhere, to go back and make sure that you spend a little bit of time on. So the 20% to 30% of individuals where these different epilepsies don't really respond to the drugs that we currently have, they may respond to other things. So they might respond to vagal stimulation, they might respond to electrical cortical stimulation, or subcortical electrical stimulation. They might benefit from having the corpus callosum surgically resected, cut in half, so that the two hemispheres don't connect, or simply being put on a ketogenic diet, high-fat diet, right? And it's a gross diet. You have to eat nothing but fat. A little bit of protein, but most of your diet all the time is fat to drive your neurons into sort of using the ketone bodies that are generated. It helps some individuals and for some forms of epilepsy. If you're really interested, I've left some of the different references here for you if you're in epilepsy research, for example. But you don't need to know all of those details, just some of the treatment alternatives that are available. But again, even there, it's not 100% effective for everyone. And so people are still looking at figuring out, how do we actually control these focal types of seizures that lead to these types of epilepsy or focal epilepsy. So since we're good at identifying the circuit, and I've just told you that everything in neuroscience is actually really all about identifying parts of circuits and modifying parts of circuits using genetic approaches, electrical approaches, etc. So you should, in theory, be able to manipulate this circuit. So I'm going to take a little bit of a detour and then get into animal models for how we induce epilepsy. And these are induction models or acquired models of epilepsy. We can inject in various places, sometimes systemically into systemic circulation, different drugs. Sometimes we have to inject it directly into the cortex. Whatever it is, we have a whole host of drugs, which tells you a lot about how it is that can actually induce epilepsy. One of these things is this idea that all of these different manipulations, whether it's repeatedly stimulating an area of the brain electrically at very high frequencies, very, very powerfully, or through chemical stimulation by blocking certain groups of receptors, whatever you decide to do, this generates this idea of kindling. And kindling is this, and this is a definition, no matter what, you should be familiar with, whether you try to memorize it for your midterm or to just know it in general neuroscience principles. If you hear about kindling, this is where through whatever means, electrical or chemical, you have actually reduced the threshold for excitation within the brain. So this threshold has decreased, which means that you're more and more likely to develop spontaneous seizure activity. And this is this idea of kindling. Now, the next few chemicals that I'm going to introduce to you are classical chemicals that either induce kindling, where they're more susceptible, or give you full blown out forms of epilepsy. So there's a whole host of drugs, and there are ones that I've specifically highlighted for you. Rodents, you can apply focally to the surface of the brain or in areas of the brain. This is what focal application means. You have to put this directly into the brain. You can apply things like cobalt. Cobalt solutions are really good at this. You can inject in tungsten acid, and it's not the acid itself. It's a gel that contains the acid. You can inject in acetylcholine as well as strychnine toxin. So for those of you who have taken neuropharmacology courses, what is the purpose of strychnine or picrotoxin? And you might have done this in one of your physiology labs, or you might have done it in 310 lab. What is or strychnine? Any ideas? So these are GABA A receptor antagonists. So in other words, you are reducing the inhibition excitation by blocking GABA A receptors through things like strychnine as well as picrotoxin. So that's if you're focally applying it to an area. So you inject into an area of the brain to be able to do this. You can also do the same thing through injection. So you can, into the bloodstream, inject in things known as, this is often done. You can do the same thing with canic acid, but with canic acid you can do it either way, systemically or in some cases in this paper, direct injection into the brain. So these can also invoke status epilepticus or animal models as well. And all of these models are really interesting. And I'm going to show you some of the results of these models, particularly the canic acid model, because they show reorganization. Now, I can't show you that directly. You're going to have to make that inference. But all of these things show that there is loss of neurons and sprouting of other neurons into the areas that are lost. The paper that I'm going to be presenting to you in Nature Communications is the canic acid model. And in this model, instead of injecting it systemically, which some individuals do, they're injecting it directly into an area of the brain that they want to be able to damage. So the pilocarpine model, I just wanted to go into this in a little bit more detail. Some of you might be interested in following up on this as part of your presentation. You might be really interested in epilepsy after this particular module, looking at the genetics of different types of epilepsy, or following up as part of your grant proposal on using a lithium pilocarpine. So the model is really simple, and it is one that you're going to see in some variation on your midterm exam. It is an important model for you as neurogenomic students to know. This is still one of the mainstays that are used. So this actually involves multiple injections. Have any of you done this experiment? I know some of you are in psychology, so some of you might have actually done this type of experiment, inducing seizure activity in rats in particular. It's a very common classic model. No? The old model actually used to be just pilocarpine. You injected in pilocarpine systemically into the bloodstream, usually through a tail vein injection, which is hard, or into another route of administration. That actually is bad. Do you know what happens when you inject in high doses of pilocarpine? So pilocarpine, and again, you should make sure that you highlight this, is a muscarinic receptor antagonist. So it is agonist. Sorry, I think I just said antagonist. It is a muscarinic receptor agonist. So what is a muscarinic receptor? So it's acetylcholine. It's one of those G protein coupled acetylcholine receptors, and this is an agonist. So now hopefully it makes sense that injecting acetylcholine might work the same way. But if you inject acetylcholine, it actually causes a lot of your experimental animals to die. And when they started doing this, some of the animals that survived had epilepsy, but it turns out that many of the animals died. So what they found that worked best was to first inject in lithium chloride. In humans, we use lithium carbonate, but in animal models, to just make the doses lower and safer and more likely for them to survive, that you first inject them with lithium chloride, you reduce the dose of the pilocarpine, and just before you inject in the pilocarpine, you also inject in scopolamine, methyl bromide, which sort of acts as a muscarinic receptor antagonist peripherally. So it helps to alleviate some of the peripheral effects, and you're hoping that the injection of pilocarpine that follows is going to cause central effects in the brain to initiate that seizure activity. As I mentioned to you before, there was a spontaneous group of rats. Normally we'll talk about mice in a few moments, and we usually talk about mutant mice, but there were a group of rats that actually showed a couple of really interesting things. It was inheritable, so it was passed generation of rats to another, and they had this generalized seizure activity. And in many ways, it looked a lot like this human absence type of epilepsy that we have been looking at earlier. And this is known as the Gares model, the genetic absence epilepsy rat from Strasbourg, where identified. So again, this is a generalized form of form of epilepsy, because it is using the model that we talked about earlier. Hopefully you all remember that from Tuesday, generalized versus focal. So again, two other mice that I want you to be familiar with, and this slide is another one that you should spend some time on. Make sure that you understand why we're interested in the genetics, because we are going to come back and talk about a little bit about it's actually a really important form of genetics. In 1957, in one of the places that we talk a lot about, the Jackson Laboratories, at the Jackson Laboratories, two different types of mice were found that had movement abnormalities as well as spontaneous seizure activity. One was called the tottering, and the other one was called the leaner mouse. And the tottering mouse was a mutant mouse that had on chromosome 8 and it was found later that the leaner mouse had the same chromosome 8 being affected. They had not just movement issues. The movement issues arose because they had these types of absence seizures as well as intermittent focal seizures. So the reason why they were always leaning over to one side or not able to move well is because they were actually having seizure activity. And it took a while for once they were actually observed to actually figuring out what was going on. Now, why are these things important? Why do you even bother talking about genetics in the first place? Don't you really want, as a neurobiologist, look at the behavior and try to figure out the circuits? Well, one of the reasons why this is important and why the different elements of the circuit are important, both of these mice with the chromosome 8 abnormality actually showed that the alpha-1A calcium channel was mutated. There was a problem with this alpha-1A calcium channel. And from this mouse model that was identified early in the 1960s, and it was identified subsequently that it was this alpha-1A calcium channel, different drug companies were able to develop brand new classes of drugs that would be able to target the alpha-1A calcium. No one had thought that a calcium channel was the underlying problem with epilepsy until these genetic models identified that this is the issue, this is the problem that is associated with this. So these are, again, this alpha-1A subunit makes up a much larger voltage-gated calcium channel and is one of the subunits, but being able to target that wasn't something that anyone would have thought rationally was at issue in epilepsy at all. So our genetic models actually do tell us something. not only about how to fix a circuit, but what things go into the circuit in the first place. So this gets back into this paper. So this is a really fascinating paper. It has a number of implications for you as young neurogeneticists. And these optogenetic techniques that we talked about in the last class, I'm going to go back into a little bit more detail. There were a number of papers, as I said. This is a 2013 paper, so about 2.5, 3 years old. But there were papers in 2009, 2010, etc., including from Carl Diceroth's group, that actually implicated that you could use optogenetics to control seizure activity. So what's new about this? Well, the thing about this particular paper that makes it really, really interesting is that unlike sort of the generalized form of epilepsy that you might get from the thalamocortical epilepsy, so it starts off in the thalamus and then spreads out everywhere in the cortex, generalized types of epilepsy. If it starts off in one area, it's just almost like random, and you can't really detect it until it's already spread, and it becomes a really big issue. And so these individuals in this paper decided, well, you know, people have published on these thalamocortical type of epilepsies before. Let's concentrate on other models for temporal lobe epilepsy. And one of the things that they did was to use one of those models that I introduced to you earlier. And this is where you can inject in canic acid. And canic acid, which I'm going to abbreviate for you as Ka, was unilaterally, only on one side, injected into the brain of these different animals, okay? So all of you probably know what area of the brain this is. What area of the brain is it? Just based on this. So this is all hippocampus, and this is what happens to the neurons in the hippocampus after you inject canic acid or canate. Can you see a difference between what you see here and what you see here on the left when you just inject in saline as a control? What are you seeing happening? Anything in particular that really strikes you? Yes, Danny?
I don't remember what missile stands for. Does it stand for?
It's cell bodies.
Oh, just cell bodies.
Mhm.
Then it just seems to be. It seems to be a wider region.
Well, okay. Again, a wider region. Okay, so you all know what these are, right? So DG is the dentate gyrus. CA3 is one area of the hippocampal formation, and CA1, the region that we often associate with sort of the output from the hippocampus. If these are all the cell bodies that you see being stained because they're nice and healthy, what do you see happening to the cell bodies over here? So the dentate gyrus is staying intact. Kind of looks like it's spreading out, right? It's less concise, it's less dark. It seems to be sort of this almost fuzzy-like appearance. And one of the things that they know is when you inject in canic acid, and it's on the next slide, that you get a sclerotic plaque being built up. In other words, there are neurons that are being affected, and there are neurons that are being lost in the areas where you have injected. And you can see clearly that there is neuronal loss in the dentate gyrus. You can see that that band that you would normally see up here with the CA1 is also lost, and there are regions of the hippocampus that are being lost altogether. If we look at fluoronissil, which is a fluorescent version of Nissil, one of the you'll see is that there are specific cell layers that seem to be more affected in the cortex as well. And in particular, layer 3, layer 4 are severely affected by canic acid injection, which is what is being shown here. Some of you are asking, how do we actually know where we're injecting all this stuff? How do you fix, like you're delivering a virus to an area of the brain? Ways that we can do this is, I don't know how well this shows up here. I'm hoping it's fairly large. You can reflect back the surface of the skull. When you cut through the surface of the skull and reflect back the skin, you can actually see the top of the skull, which is what is being shown right here. And if you look very carefully, you can actually see the different suture lines. You can see lambda, for example, and if you're looking very carefully, you can actually see another suture line where different bones on the skull connect, which is known as bregma. And what this is showing you is that relative to bregma, we are moving 2.5 millimeters back, or 2 millimeters back, or 1.7. So we have precise surgical coordinates to be able to do this. We can do this when we have these stereotactic frames that allow us to target specific brain regions. Okay, so we inject in this canic acid. It's unilaterally into the dorsal hippocampus, as I've just shown you here. It shows loss of neurons, and it shows sclerotic plaques being built up over time. And this model also results in spontaneous seizures that arise near been damaged. There is, again, very little or no behavioral manifestations early on, and it's only later that after about 3 to 4 weeks or so, they have to wait, and then they start to see spontaneous recurrent seizures. You don't get seizure activity right away after you inject canic acid. You have to give them a little bit of time, a period of weeks for this to develop. So neurons die, the sclerotic plaque builds up. It's just like what happened with HM. HM had exactly the same thing, and he had to have that sclerotic region in the hippocampus actually removed. I don't need you to memorize things like stage 3-4 on a Racine scale, etc., but they were able to eventually see these types of seizure activity. Again, no need to memorize this. It's a brilliant piece of engineering. If our brain is a circuit, build a circuit on top of a circuit, right? Which is what these engineers decided to do. So what they decided to do was to put in electrodes that would measure activity in the temporal lobe, see if there was any hint that there was going to be epileptiform activity after a few weeks after canic acid injection, measure all of this on an EEG, put it into a digitizer, which then allows you to see, you know, suddenly we have this onset of what looks like spiking. It looks like you're going to have temporal lobe epilepsy. Also have expressed in an optogenetic circuit to turn it back off. So once you start to see that the neurons are becoming too excited, you have a circuit that automatically recognizes that a light should flash on to turn the genetic circuit that sits on top of this. Is that okay for everyone? It's a really brilliant idea. It's a simple idea. Build a circuit to control a circuit.
It reminds me of like noise canceling headphones.
Yeah, it's kind of like noise canceling headphones on a very, I guess, invasive way. Okay? So again, here's the model, and I want you to think about this model just a little bit. Okay, and some of you will be able to pick up on this, and for some of you, you may need to come and see me and go over this in a little bit more detail. So this paper was published in 2013, and in 2013, papers came out, and they had developed rats already that contain what are known as driver lines. So they had one group of mice that in every neuron that contains this particular gene, CAMK2, it expresses Cre recombinase. Every single neuron that has CAMK2 also has Cre recombinase. They also developed in every single neuron a halorhodopsin mouse line that also was Cre dependent. Okay, think a little bit about this. Okay, so they have a halorhodopsin mouse that contains the gene for halorhodopsin. Never had it before. Now every single neuron in this mouse has halorhodopsin associated with it on the right, with a Cre driver line that is the Cre recombinase is expressed in CAMK2 expressing neurons. So remember, this means that if you cross this and it works, and their offspring will then express halorhodopsin in excitatory neurons. Okay? How does that work? Yes?
Well, I'm assuming that, so one mouse,
Yeah.
have all Cre in their neurons.
Yes.
However, they don't have the Cre-dependent halorhodopsins. So
Sure.
when you cross it with the halorhodopsin mice, then their offspring will have both the CAMK2 gene as well as the Cre-dependent HR.
So their offspring have now Cre recombinase and excitatory neurons and halorhodopsin, right? But why would you cross this? Doesn't Cre recombinase actually excise things? Doesn't it cut things out?
Only with LOX-P in them. So
but this Cre dependent means that it's got LOX-P sites. So how would that work? So it has LOX-P. How did you engineer it so that with recombinase that now you're going to get halorhodopsin? Recombinase cut things out of, doesn't it cut things out, excise it? Yes, there are two possibilities, by the way.
So is that you have, I would imagine you could be using Cree to cut out that stops the transcription of the,
Okay.
the making of the protein.
Okay.
Or you can be flipping something to turn on a promoter.
Perfect. So you do understand that. So one way is to have inverted the sequence of halorhodopsin, and you could have done it that way. Much more difficult to do in transgenic lines, or to have a stop codon, which is being floxed or removed through Cree recombinase. Is that okay for everyone? Kind of yes, kind of no. If you don't understand it, then you can come and see me, and it's really more like X's and O's and diagramming this out. They cross these mice, they inject with canic acid. These mice now all have halorhodopsins everywhere. You don't inject in halorhodopsin. You don't need to use a virus or anything else. Here with canic acid, these seizures emerge over the course of a couple of weeks. You then implant in the fiber optic cable, which is what the implant is showing. And then you flash on orange light, which is what this little orange bar means. So because this is hooked up to an electrical circuit, every time the mouse has a seizure activity, which is what is being shown here, if you flash on the yellow light and you allow it to flash on, the seizures stop. If you let the seizure activity go, which is what you're seeing here, and no yellow light flashes on, the seizures go on for a while, which is what you're seeing here. So every time this little orange light comes on, seizures stop. Every time you just let the seizure activity that you've detected go on. So here it's going on in E, and it continues on over here. Is that okay for everyone? Yes, Danny?
I thought that we talked about in the last class that I think it's SSFO.
Yep.
You don't need to keep the light on. Is that the same one they use here? Or did
That,
they use...
that one is a 2014 creation, so
yeah.
not yet published at the time. So you don't have to really go nuts on this. The basics if you want. And when you're reading a paper, there are certain things that you should be taking a look at. When you see something like floxed, it just means that it's undergone a Cree-Lox recombination event. So what has been floxed or removed by Cree recombinase is this stop codon, as you have mentioned. So this stop codon that you see here, I'm hoping that it shows up. Let me just see, I can't see well here. Actually not sure that it is on here. However, it is a stop codon, and it's the stop codon that is actually produced in triplicate, and that's the major thing that you'll have to worry about. Danny, as in to answer your question from before, these are the specialized LOX-P sites to prevent it from flipping back and forth, the LOX 2272. And again, just for your own information. So this is how they did it. They have halorhodopsin. Normally it doesn't get transcribed because there's a stop codon in front of it. But now with the LOX-P sites around the stop codon, it gets removed. And when that stop codon gets removed with crossing with the Cree recombinase mice, then it allows for the expression in those CAMK2 groups of neurons. So remember, these are excitatory neurons. And now you have this sort of feedback loop where when you turn on the orange light in response to increased activity, so the activity is going, these mice have halorhodopsin in yellow light, all of that activity stops right away. If you don't stop it with an orange light, then the activity continues on, and it's really high frequency activity. It is epileptic behavior. Richard.
Is there a specific reason why you need to have created LOX-P in the first place? Doesn't that seem kind of like an extra step? Couldn't you just get your halorhodopsin into your CAMK2 another way without?
You could. So here you don't have to do another injection. That's one, right? So you just cross it, you let the mice grow to adulthood, then you do the injection. Here you're making sure that they're only expressed in a certain subgroup of neurons, excitatory neurons, and this will become important with the next experiment that I show you as well. So from this slide, do you think their experiment worked? Can they have a feedback loop where when they see spontaneous activity in their canic acid model, that they're able to turn off epilepsy? Yes? So again, all of the data over here on the bottom panels, which is showing you that in the presence of light, when they have light flashing on, overall it's stopping within seconds. Okay, so the seizure activity stops whenever this optogenetic switch is turned on. And overall, when you take a look at the amount of time that the seizures last for, it also goes down dramatically. So did it work? Absolutely. Was it great? Also a really powerful experiment. Do you like this experiment? Yeah, it's okay. It's only a Nature paper, right? Think, okay? I really want you to think about this for a second. I showed you this at the beginning. Where are your excitatory neurons? Your excitatory neurons are in the dentate gyrus. They're in CA1. They're everywhere, right? Do you still have those? I told you it's a sclerotic plaque. So how are they seeing their effects? The excitatory neurons that contain CAMK2 have been wiped out with canic acid. After canic acid, you get spontaneous seizure activity. Optogenetics seem to be doing something, but why is it doing something? It's a tough question, and they don't have a really good answer. So even if it's published in Nature, think critically, like think really hard about the types of experiments that you're actually seeing. So I'm just going to take you through this really quickly because I don't need you to memorize all of this. It's something that I hope that you've heard of. One of the advantages of using halorhodopsins, because this goes back to Richard's question, you are turning off the activity of excitatory neurons, the ones that are being overly excited after canic acid, overly excited in epilepsy, off the excitation. So what are the advantages of using halorhodopsins? One advantage is that this all uses sort of a long wavelength or red, okay? And this orange or red wavelength of light, in theory anyways, if we continue to redshift the opsin farther and farther along, right? So what comes at the end of red? What's on the other side of red? Infrared. So can you see infrared? Because if you could, you'd be kind of going nuts because everything would be all hazy. You can't see ultraviolet, you can't see infrared. However, at some point, we might be able to adjust opsins even further, redshifting them all the way so that they can be turned on by infrared light. Okay, why is that important? I don't know if you've ever FNIRS. FNIRS is actually a really interesting. Anyways, FNIRS, or functional near infrared spectroscopy, is kind of like fMRI. And what happens is you wear this band. It's very easy to actually do. And this is the band that you put on. And it shoots in infrared light through your skull. Directly through your skull. It goes in at about 3 centimeters or so. So it penetrates fairly deeply all the way into the cortex. And what it does is that it actually shines infrared light, and it measures the oxygenation state. So is your blood oxygenated or deoxygenated? And because oxygenated and deoxygenated blood have different IR frequencies, about is this tissue more or less active? It's like functional magnetic resonance imaging without the MRI portion. Okay, all it's doing, end of little red lights that is constantly shooting red light into your skull. Because it's infrared, it can penetrate deeply, and then it can also detect the reflected red light back. This is just for your own information. I'm not going to be testing you on it. In theory, this is actually a really useful technique, but in practice, it's not something that you do or hear about very often, either in psychology or in neuroscience. And there are lots of reasons for this. So it's good, it's fast, it's cheap, it's mobile. You don't have to sit in an MRI device. It's better than EEG in some ways, but we don't tend to use this very frequently. Okay, so I'll kind of leave you with that for the time being. Maybe down the road, if all of this works, and there's an easy way to introduce in a opsin into someone's brain, you don't actually have to implant an optrode. All you need is a little band of these optodes, which shine orange or red light, and they could stimulate that whenever a seizure activity turns on. So as Richard pointed out a little bit earlier, one thing that happens is you could turn off excitation. You use halorhodopsin's quiet down excitation, but they all could be the other way. So there are also GABAergic neurons in the areas that we destroyed. Those GABAergic neurons might be the ones that are actually surviving better anyways, because they don't get as easily destroyed. We can mark these GABAergic neurons because that is unique to inhibitory cells called parvalbumin. And what they have generated are these parvalbumin Cre lines, just like we have for CAMK. These represent less than 5% of the neurons. So if we took a look at this, because they have so many connections to so many different cells and they're inhibitory, can we also control epileptic activity just by inhibitory neurons? Less than 5% of the population in the area took a look at. And sure enough, when they cross those parvalbumin containing Cre lines, and now they need to excite those Cre by having a stop codon rhodopsin. So wherever channel rhodopsin is expressed in inhibitory, when you stimulate those, what happens is exactly the same. So turning on, using channel rhodopsins, the inhibitory cells, even though they're a really small population, what happens there is that seizure activity, duration, as well as how quickly they get turned off, also disappears, suggesting that either method, stimulating the inhibitory or decreasing activity of the excitatory, is equally valid. So I want you to be familiar with both methods. If you're asked how you might design or follow up on this experiment related to Cre driver lines, it might be something worthwhile to make sure that you understand. Again, I'm not going to do more than just mention this because this is actually kind of interesting. The gist of all of this down here is this, and they were worried about the same things that many other scientists ahead of them were worried about, is if you keep shining light on these guys, and this is, I think, your question, like why not use the stabilized opsins? That's the bottom line. But if you keep stimulating these things, do they actually get effect? And what they found is, the bottom line, when we check for activity in neurons, one of the things that we look at is to stain them with a protein known as CFOS, which is a gene. Anytime the activity goes up, we should see more CFOS staining. But what they found was we don't see much more CFOS staining unless we, which is slide is saying, which is really what I want you to do. If you want to go through and read it, that's fine. You don't have to memorize these numbers. But what this is, when you stimulate using halorhodopsins or channel rhodopsins, as least detected by CFOS staining, which is a measure of activity, we don't really see it until we start to use a lot of stimulation at the same time in their experiments, okay? So again, just for your own information, so this is the most, see, we don't have a really good way to treat it right now. It's really pie in the sky, but to be honest, I happen to know this for a fact, that this will be sort of the next clinical trial that is done. Right now, the clinical trial in humans on optogenetics is for chronic pain. One will actually be on epilepsy as a therapeutic sort of intervention related to this. So I think one of you were asking, will you be posting some more guidelines work? Yes, I will, and starting this weekend, idea. I'm not a big fan of these types, quite honest, because I sit in the office, I get to hear a lot of these discussions, and it's, if I meet these criteria, but the reality is, hearing all these discussions, it's still, actually it's more subjective using this type of rubric than not using this type of, if you meet, like, you get 4 points if you this. So what I suggest doing instead is that I'm going to post for you a sample of what I consider to be an A with comments, why it got an A, versus something that did not get an A with comments as well. And I'm hoping that by reading and comparing those, you'll have a better idea of how you on your summary assignment, as well as on a group grant proposal. Is that okay for all of you? So I material. Last dense lecture for the midterm. The next 2 lectures are actually I'd say more neurology based than anything else, and we'll come back and talk about this again on Tuesday.